Hello, This is Mrs. Rowe, and welcome as we continue to learn biology from home. And so, this part here in Chapter 5, we are going to talk about quickly the cell cycle, the chromosome structure, which is so important to how cells divide as we look at asexual reproduction and mitosis. We're going to do a flyby of mitosis. We're not going to worry about the details. So, cells need to reproduce. That's one of your traits of life. If they can't reproduce, then when cells die, that would be the end of the species. And cells also need to reproduce, as we talked about yesterday, for growth and development, for repair and maintenance. We lose millions of skin cells every day as those skin cells are getting um, lost and damaged because of all that exposure at the surface. We need to be able to replace them. Our blood cells are being replaced every 3 months. So certain cells we're repairing and replacing very rapidly. Other cells, such as our nerve cells and our muscle cells, not so much. So all cells have a different rate at which they are being replaced. The key vocab here is to be what is the difference between DNA, a gene, and a chromosome. So three important vocab words that people tend to mix up, and we'll come back to this at the end. Couple more vocab words. Histone. Histone is simply a protein. I think of it as when you think of a spool of thread, the wooden spool that you wrap the thread around, that's like your histone. It's to kind of just keep things organized and help keep it all tightly intact. When you think about each cell has 6 feet of DNA, that's all the hereditary information that is in every single nucleus of every cell in your body. How do you pack 6 feet of DNA into one cell? Well, you got to have a pretty good packing system, and histones are part of that packing system. Some people might say, how can a tiny cell that you can only see under the microscope have that much DNA? Well, it might be 6 feet mathematically calculated in length. However, its diameter is only a few atoms across, so you absolutely still can't see it, even though it's that long. Even in a regular light microscope, you're not going to be able to see that DNA. Okay, so as we go further, and we're just going to start with the cell cycle, and we've talked about this already in some of our reviews. So the cell cycle, and I just want to come down to, which one did I just have ready? This one, I think. That's not the one. But we'll use this one. This one looks fine. So we have interphase, our G1, SG2, where our cell is a normal working cell. You don't really see anything happening under the microscope. But G1 is really about the growth, because you have two new cells that are smaller. They've just divided and they're going to grow. But from the moment they divide and their chromosomes unwind back into chromatin, that's back to a working cell. If it's a pancreas cell, it's producing insulin. If it's a skin cell, it's flat and it's protecting and doing its job. Over here, and I have another one that we're going to come back to look at the checkpoints. If that surface area to volume ratio starts to drop because the cell's starting to get too big, if there's some other trigger, like there's been damage and we need to replace the damaged cells, or we're growing, but there's some sort of signal happening that says, okay, we need to go into cell division. Now, this period of G1 we mentioned yesterday could be a few hours, could be a few days, could be weeks, months, or even years. It all depends on the cell and what is happening. Obviously, if you're an adolescent, your cells are going to be dividing much more rapidly than somebody my age who is no longer growing. And then S phase, really important. This is the one you want to pay attention to. That stands for synthesis. That is where the DNA is copied, because before you can divide your cell into two, you've got to give each cell a copy of the DNA so that it can continue to do the jobs with that. And then it will continue to grow, and it's going to get its whatever spindle fibers, organelles. It's going to just start doing final preparations of getting ready. There are checkpoints along the way. So if we come back over to here, this one shows some of our checkpoints. So G1, is your cell big enough for it to divide? Is the environment favorable for cell division? And then once it goes past this checkpoint, this is like a stop sign. Stop, check, and then continue on. And these checkpoints are actually proteins that help regulate the cell cycle. So these proteins are therefore coded by your DNA. There are genes for these checkpoints, and that gets important when we talk about cancer. So in the S phase, we copy the DNA, getting ready to divide, and then here's another important checkpoint over here. So this is all part of cell regulation, so that cells don't divide too quickly or not enough. And a really important piece in here is also, there's a check to make sure that your DNA is copied properly, that you don't have any mutations or damage to your DNA. As a matter of fact, when you get a sunburn, what you've done is damaged your DNA, and so the cells will self-destruct and your skin starts to peel because the damage is beyond what the cell can repair. So if everything is good, then it's going to go forward into the stage of mitosis, and mitosis is strictly about dividing your DNA in the nucleus. And remember, all of this cell cycle is for a eukaryote. We're going to start talking about prokaryotes tomorrow. And it's going to go through what I call PMAT. Phases of mitosis are terrific. That's all you need to know. There are 4 phases. There are 5 phases, G1, S, G2, MC to cell cycle. There are 4 phases to mitosis. So please try to keep those separate. So we're going to look at that after. And then after the nucleus divides, it goes into what's called cytokinesis, and we get 2 new cells. So let's come back down to this one, where it shows you part of the mitosis here. They're lining up in the middle. We talked about that briefly. So there's your prophase, metaphase, anaphase, telophase. So just be aware and recognize. I'm not going to expect you to know exactly what happens in each phase or the details. I just want you kind of be able to recognize that. And then cytocell. There's 2 ways the cells can divide. If it's an animal cell, there's no cell wall. It's going to be like a belt of proteins, kind of looks like a snowman. It's just going to pinch across the middle. If it is a plant cell, you're going to need to form what's called a cell plate. So that cell wall is going to start to develop across the middle. And once that cell wall or cell plate grows across the middle, then the cells will finish dividing. But it's a little bit different in a plant cell versus an animal cell. And finally, what are these little X's? These are our chromosomes. So we wanted to talk about chromosome structure really quickly. I asked you to make a 3D model. 3D model is just whatever you have around the house. I'm looking at creativity and can you apply the vocab. So this was the 3D model I came up with. I happen to have these nice big spools of thread, and I wanted them to be the same because the 2 halves of my chromosome are identical when we put this together. So all cells, before they go into S phase, only have one of these. And when they have just the one, it's still called a chromosome. We don't have chromatids until S phase when they've been copied. This is still now a chromosome, even though there are 2. So it gets a little confusing. So this long and unwinding stringy part, I could use it to represent my DNA. It all depends how I want to do this. I decided to make this my chromatin. Do the best I can. It's very slow. So that's, and I think of chromatin, my long unwound DNA, that is the state that it is in during interphase, because you want to be able to get at the genes. The genes are pieces of DNA, segments of DNA that code for protein. So you have all these genes, thousands of genes along each strand of DNA for each chromosome. And then the DNA wraps around these histones. So this, I'm just going to do H for histone here. I have the spool representing the histone that the DNA, so I guess really this around the histones is the chromatin. So I guess DNA would be better here. This whole thing together is the chromosome, the 2 of them. The center here, centromere has center in it. That's just a protein that holds the 2 identical halves together. So in S phase, my chromosome was duplicated or copied. This is the identical copy. Each half is called a chromatid. A chromatid, identical kid sisters. We're going to use our twins to make some kid cells. Okay, so chromatid. This is a exercise in patience. Okay, there's your chromatid, the individual one versus the chromosome. And this is important to have these words down because in mitosis, we're going to break the centromere and we're going to give each cell is going to get 1 chromatid. So each cell gets an identical copy of the DNA when it divides. So finally, let's take a quick flyby at mitosis. And this is what we were talking about. What's really cool is when you are looking at these cells under the microscope, here's interphase, you can see what's happening in mitosis. It's visible under a light microscope. There's some dramatic changes taking place as the cell is dividing. So basically, as we go through in interphase, we have our chromatin. You can see a nucleolus. We have an intact cell membrane. Again, this is for eukaryotic cells only. Prokaryotes do not go through mitosis. So this is interphase right here, and I'm not going to spend the time trying to write all this out. And then when we start to see, so we did this yesterday, prophase with our hands crossed. This is why we crossed our arms in an X shape. This is where the chromatin winds up into chromosomes as it packs up around those histones, the centromere keeping them together. So there, when I see the X's, I'm in prophase. I'm just beginning. This nuclear membrane that was around the nucleus has also, you can see the remnants of it here, is disappearing and spindle fibers are forming. And those spindle fibers are what going to pull these apart. They don't just magically split. It's these fibers that attach and help pull it apart. And so then we go into metaphase. So phases of mitosis are terrific. Metaphase, they line up in the middle. Again, it's the spindle fibers pulling them into the middle. And there's a spindle fiber attached to each chromatid. This isn't showing the attachment. And then the spindle fibers shorten and pull the two sides apart. And once our nucleus has divided so that each side now has its, now that they're in separate cells, these are actually called chromosomes again. Each cell now is back to its regular number of chromosomes. The nuclear membrane reforms and cytokinesis starts to happen. So cytokinesis tends to overlap telophase. I say telophase is the end. We get, or T for two nuclei reform in telophase. So again, you do not need to memorize the steps, but just be aware that there is a 4-step process to dividing the nucleus. And that is it for now. So mitosis, asexual reproduction, it's how we make more of the same. If we have a cut and we want to repair our skin cells, if we are growing, that's the sort of places where mitosis is going to come into play. So with that, you should be able to answer, what is the difference between DNA, genes, and chromosomes? Which is the smallest, which is the largest? Well, obviously chromosomes are the largest. It's all the DNA all condensed and wrapped up. And genes would be the smallest. Genes are just segments or pieces of DNA. They're portions of DNA that code for a protein. As we look at that, I hope that helps, and have a great day.